Okay, so firstly, thank you all for listening, and thank you to the organisers for giving me the opportunity to speak here today. So today I want to talk about some of our recent efforts to use deep learning in NMR analyses. And I want to start off by thanking my group. So I'm a postdoc in Fleming Hansen's lab in UCL in the UK. So thanks to Fleming for getting me on this project and all the help he's given me. Thanks to Harry, who recorded some of the spectra I'll be showing you today. Vibav has helped produce some of the samples and recorded further spectra. And then thanks to the rest of the group for really useful discussions. Here's the group in kind of pandemic times. And thanks to the BBSRC for funding this project. So I'm going to give you, because this is a very quick talk, I'm going to give you a one-slide overview of kind of some of the ideas of deep learning. So it's a subset of machine learning, and it employs these things called deep neural networks, which are very loosely inspired by how animal brains function. So the simplest type of neural network you can get is this thing called a multi-layer perceptron model. And you see these nodes which are arranged in layers. So you have an input layer, you have some hidden layers, and then you have an output layer. So I've shown two hidden layers here. You can have as many as you like, can have as many nodes as you like. And it's for depth in the hidden layers that leads to deep neural networks. The beauty of deep learning is, and supervised learning specifically, which is what we're going to do here, is that you give your network an input and you give it an output. And basically, it has to figure out for itself how it's going to map said input to the output. So it's not concerned with your preconceived ideas about how you might want to go about solving the problem. And that can lead to like some creative solutions. And the reason we get excited about deep learning is because you might have seen things like this. So last year, DeepMind's AlphaFold 2 kind of solved the protein structure prediction problem using deep learning methods. And we know that deep learning methods have now shown superhuman performance in a range of activities. So we're not quite there yet in NMR, but quite a few groups I'm aware of are now trying to explore how we can use deep learning in NMR and also magnetic resonance more generally. So I want to talk about two applications we've been working on quickly. So firstly, how we can reconstruct non-uniformly sampled data. And secondly, how we can virtually decouple spectra. So we don't use an MLP model. We actually use something a bit more complicated. I'm not going to dwell on it too much. So this is our architecture that we use that we call FIDNet. And the crux of it is this. So if you want to analyze FIDs, the information FID is not localized to a particular kind of point in that FID. It's spread out through the whole thing. So you need a way of looking at both short-term and long-term patterns within an FID to learn information on the resonances. And you need kind of an efficient way of doing that. So we borrowed an idea from DeepMind. They have this WaveNet architecture for, it's a generative model for raw audio, and we borrowed some ideas. The key idea we borrowed is this idea of dilated convolutional layers. So we basically train these filters to pick up features from our FIDs, and then we train bigger filters which have holes in them to learn kind of more long-range patterns, and we can stack these all on top of each other to get an overall picture of what's going on in a particular FID in an efficient way. And there's a few more complications in our model. We have things like skip layers and gated activation units. It's explained a bit in our ChemArchive preprint, and I'm happy to answer more questions about that in the breakout room. But the idea is we can train this to do useful things to FIDs. And the first thing I want to talk about is non-uniform sampling. So I'm sure a lot of you are familiar with this already, but a quick overview. So say you have a 2D spectrum and you Fourier transform it in your direct dimension, you're left with this kind of interferogram where your time domain is still in your kind of indirect dimension and your direct dimension has been Fourier transformed. You do a second Fourier transform and you get your nice spectrum. The thing is here, with your fully sampled spectrum, each complex point in your indirect dimension means you have to record 2 more 1D spectra. So it's quite time intensive. So what you can do is think, I'm not going to record all of them, I'll skip some of them out. And that gives you a non-uniformly sampled spectra. And the points you sample are defined by a sampling schedule. If you try and Fourier transform that, though, you're going to end up with a bit of a mess. So what you need is some way of reconstructing the points you've missed out to get like a reasonable spectrum at the end. And ideally, you know, the perfect spectrum. And there are a few algorithms out there which do this quite successfully already. And but we wanted to see how our approach would compare. So specifically what we can do with FIDNet is we can't reconstruct a whole spectrum at the same time, but we can split it into chunks. We can reconstruct all of them at the same time. We can use the kind of parallelization power of GPUs to do this quite quickly, and then we can stitch the spectrum back together, and then we can Fourier transform it. So I'm going to briefly show you some examples of the kinds of things we can do with FIDNet. So what we find is it's really good at reconstructing very sparse data, so situations where other programs may struggle, and it's quite good when your data is slightly noisy as well, and it's also very tolerant to the kind of sampling schedule that you use. So this is a reconstruction of ABP1P, which is an HSQC. So we're reconstructing it from 15 points out of 120. So you're getting this spectrum in an eighth of the time. Something a bit more challenging with more peaks is this T4L sample. Here it's doing a very similar job to SMILE. HMS IST does a good job, but it's very reliant on having a plus on sampled sampling schedule. And then here's an HDAC8, so this is a methyl-trosy spectrum, and here's the FIDNet reconstruction. So one thing we find in general, if you go into why the RMSD is what it is, in general, our network gives you less false positives and false negatives, but it's slightly less good than other algorithms at kind of exactly getting the intensity and positions of peaks right. One other thing that falls out of our kind of analysis is you get an insight into the confidence of your reconstruction. So for example, in this ABP1P spectrum, you can see these red points indicate kind of uncertainties. So we can identify a false positive at the bottom of this spectrum quite easily, which is really useful. And similarly, on the HDAC8 spectrum, we can use this kind of approach to identify two false positives. So another thing I want to talk about quickly is how we can use FIDNet for virtual decoupling. So commonly, especially when you're doing direct detection of kind of carbon-13, you'll have a homonuclear scalar coupling that will evolve during acquisition. Experimentally, you get rid of this using something like an IPAP or spin state selective scheme, and that works well. The problem is it doubles basically the phase cycle of your experiment, because you have to record both an in-phase and an anti-phase spectrum and take linear combinations to get your result. So can we do something like this to go straight from the in-phase to a decoupled spectrum? So we first wanted to apply this to CC correlation spectra. So this is something that was developed by Ruth, who was a previous PhD student in the group. And when you have this kind of motif where you have a terminal carbon and a penultimate carbon with distinct chemical shift distributions, you can record these kinds of spectra to get insight into protein side chains, but you have to use an IPAP scheme. So what if we trained our deep neural network to do this? What you find is it works basically just as well as the full IPAP scheme, but it only uses the in-phase spectra. So this is the valine spectra of T4 lysozyme, and we can get really good decoupling from the in-phase spectra alone. And the other thing is the network's trained to denoise as well as decouple. So the final kind of spectrum you get from FIDNet is basically as good as the IPAP spectrum, but you're getting it in half the time. So you can do the same thing with the isoleucine spectrum of T4 lysozyme. You can see the input is fairly noisy, and the output is quite well denoised. We can apply the same tricks to con spectra. So these are really useful for looking at IDPs in particular, where you get rapid proton exchange with the solvent, which might broaden your signals beyond detection. So For ubiquitin, so NMR talk, so you have to include ubiquitin. You can see we get really nice decoupling, even when you have quite overlapped peaks. It's not really a big problem. We recover all the peaks we expected to recover, including quite low intensity ones. And we can do the same thing also on alpha synuclein, which is a very well studied protein IDP by NMR. And you can see we can effectively get really nice spectra in half the time on the basis of the in-phase spectrum alone. So you don't have to change your kind of experiment at all. You're running the same thing you would run. This is just a post-processing thing. The last thing I want to show is how you can use this in HNCA and HNCOCA spectra. So here you get a coupling in the carbon dimension because of the C alpha, C beta coupling. That kind of limits your resolution. Okay. So in this case, the challenge is that everything has a C alpha C beta coupling except for the glycines, and those we want to leave kind of unmodulated, whereas everything else we want to decouple. And you can see the glycines are kind of left alone, whereas we decouple everything else quite well, even the very low intensity peaks. So here's a quick summary of what I've shown. So we can use this architecture for both reconstructing non-uniformly sampled spectra and virtual decoupling. And yeah, so the code for doing this is available on GitHub, so you can reconstruct the spectra in like one line. We've got a pre-trained kind of network which you can download, and if you have any problems, well, you can get in contact with me by these means. And yeah, thank you for your attention, and I'm happy to answer questions in the breakout rooms afterwards. Thanks.
That was fantastic. Thank you very much indeed. So let's pass on to our final speaker, Aaron. If you want to get started.
Okay, can you see the cloud?
Yep.
Cool. Okay. I'm Aaron. I want to thank you, the committee, for inviting me to give this talk. This is a talk about my PhD project, which is about to finish soon, maybe, hopefully. And it's about enzyme activity regulation beyond allostery, substrate discrimination via protein switch. I work with the enzyme beta-phosphoglucosamidase from lactocase lactis. It's a monomeric enzyme, 2 domains, 1 active site in the middle, that catalyzes isomerization between beta-glucose 1-phosphate and glucose 6-phosphate via a glucose biphosphate intermediate. In the steady state of the reaction, glucose 1-phosphate binds to the phosphate sugar species. The enzyme transfers the phosphate, generating the biphosphate sugar intermediate. This one is released and binds in alternative orientation, and the enzyme takes back the phosphate, reconstituting the phosphoenzyme and generating glucose 6-phosphate. It's been reported for this enzyme that the activity profile is very different depending the phosphorylating agent you use to generate the phosphoenzyme. If you use the intermediate, you get linear kinetics as any other classical enzyme. However, if you use acetophosphate or fructose biphosphate, you get a lag phase, which is very common in hysteretic enzymes. So we thought that this is maybe a mechanism of post-radiation control of enzyme activity in PEM. So we want to look at this further, looking at the target of the phosphorylating agent, which is substrate-free PEM. So when we look at this enzyme solution, we see 2 species that we name conformer A and conformer B in a slow exchange. When we estimate the exchange rate is between 1 second and 5 minutes, it's quite slow, and they're basically near equal populated. The chemical sheet difference between the 2 species is localized in the core domain, very close to the active site. This chemical exchange is due to cis-trans protein isomerization. When we generate the variant P146A, we see only 1 species in solution, which is a good model of conformer B, as you can see by the chemical sheet difference between the variant and conformer B, in contrast with comparison with conformer A. So now we have a good model to look at conformer B and what's the involvement in a catalytic cycle. When we look at the structure by X-ray crystallographic, we see that the general features are basically the same. The only drastic change is the position of the side chain K145. In wild type, when the proline is cis, K145 is engaged in the active site, and it's really important to provide charge balance in order to transfer the phosphate between the sugar and the 8. However, in P146A, where the alanine is trans, you see that K145 is actually pointing toward the solvent and is away. It cannot provide charge balance. So we think this has implications for activity. Maybe P146A is a really bad enzyme. However, it's not. It's actually quite good. This is the general features of the wild type activity, and this is the P146 activity. You can see the same behavior depending on the phosphorylated agent. However, you can see activity. When we did a more exhaustive analysis using glucose 1,6-biphosphate as a phosphorylated agent, we see that the effect of the mutation in the catalytic process is in the catalytic rate, which goes 20% fold decrease, and in the Km of GBP, which has a 20-fold increase. The G1P steps are not affected by the mutation. This is interesting. This means that conformer B is actually involved in the steps that also require GBP. So is conformer B active or is not? When in the lab we have a method to study transition states of phosphorus transfer reactions using metafluorides. We generated a beta PGM magnesium trifluoride G6P complex of both wild type and P146A, which is a good analog of GBP glucose biphosphate binding. What we saw in the structures, crystalline structures, is that they look basically the same. They look so much the same that there's a cis alanine as well as cis proline in there. A cis alanine is 1,000-fold stabilization of our really high energy conformations, but PEM just doesn't care. Just want a cis alanine in there, and it has a cis alanine in there. That's really also interesting. This means that conformer A is the most active form in PEM, and conformer B is an inactive form. The mutant is active because it can go to adopt the conformer A when GBP is bound in there. So we come up with this kinetic model to explain the kinetics, the lag phases. Basically, you have the usual catalytic cycle that I told you about. We have phosphoenzyme that binds G1P, generates GBP, releases GBP, binds, and transfers the phosphate back to generate G6P. But also you have the steps that involve B. Basically, you have the B species right here that can also bind phosphorylating agent, and the lag phase depends of this rate right here in the interconversion between B and A. So bad phosphorylating agents will actually have a really slow rate of interconversion and generate a lag phase, and very good phosphorylating agents as GBP will have a really fast interconversion rate. We wanted to test this model by looking at the species phospho B. If the species phospho B exists, then we know that this model is correct. The problem with that is that the phospho B species is transiently populated to attack in the first 20 minutes of the reaction, or even sooner in using an acetyl phosphate. So we come up with this device to do real-time NMR with fast mixings using the magnet, with a dead time of 2.5 minutes and a first time point of 2 minutes after that. So we have a way to do a structural analysis the first 5 minutes of the reaction. It's very fun experiments to do. This is a close-up of the trophy, looking at one of the residues that is a really good reporter of what's happening in the active site, alanine 113. So basically in substrate-free, we have two species, A and B. In P146A, we have only one species, which is B. If we add fructose biphosphate, we have three species in wild type. One is phospho A that we know because we have a metafluorine analog that we can compare to. There's a A fructose biphosphate complex that we can assign because we also have an analog of glucose biphosphate binding. And there's also phospho B complex, phospho B species right here that we know because we can also see that in P146A. Most importantly, when you add GBP, you get a conformer A GBP complex in wild type, very, very broad. But in P146A, you don't get phospho B at all. You get a P GBP complex or a phospho A G6B complex. that means that glucose biphosphate is able to push the equilibrium towards A and assist alanine, which is really, really nice and neat. So we know that's true. We know we have a phospho-B species that is present when you add fructose biphosphate, and you cannot see it when you add glucose biphosphate due to the ability of the enzyme to promote that isomerization. So we come up with this general mechanism. So in opposition to allosteric, in which you need an allosteric effector and another molecule that will activate your enzyme, we don't have that in PEM. We don't have another binding site. In allocarry, there's a competition between an exchange rate and a catalytic rate that are very similar in magnitude, which don't have that. Our exchange rate is very slow compared to the catalytic rate. So we are proposing a new mechanism called allomorphy, in which we have an exchange rate between two different enzyme conformations, which is very, very slow. And also we have alternative kinetic pathways depending which activator is present. If we have a really good activator, it will go through the fast route, and the K-cat you see is the true K-cat of the catalytic cycle. And then you have a very bad activator, in this case fructose biphosphate, you will go through the slow kinetic pathway, and the K-cat you see is actually a conformational exchange rate. It's not the true K-cat, which is very neat. This was published not so long ago, so if you want to know more about it, it's a very neat paper, if I may say so. So now I want to talk about what's next as the end of my PhD. Now I want to know about more the structural determinants of allomorphy, how GPP is able to promote this isomerization and FPP is not able to do that. So for that, I'm using the D10N variant and the double mutant D10N P146A. We know that D10N is able to form stable trap complexes with glucose biphosphate, as we have published that already. So now I want to use these models to study how these complexes here look in the model and see what's happening there. And in this the latest news, don't tell my supervisor, he still doesn't know. I successfully crystallized a D10N FPP complex, which with a very, very neat fructose biphosphate in the active site. You can see this hydroxyl right here, which is particular for the sugar, it's not in glucose. Biphosphate. In this crystal, you can see the lysine is engaged in the active site. The proline is as we had expected for this molecule. So now I want to do this with a double mutant in which I would expect the lysine to be outside and the alanine to be trans for the fructose biphosphate complex. And it will be in, and there will be a cis-alanine in the glucose biphosphate complex. Thank you. I want to thank everybody that has participated in this project, particularly Henry and Nikki. This has been a team effort. I'm very thankful for that. Thank you. That's all.
Okay, thank you very much.